by MBSI. And the whole purpose of this meeting is to share the knowledge as well as the topic that is assigned to that day, so that student participants as well as researchers can share their ideas, or if they are in some trouble, they can also exchange those things. And few experts who have joined this session can also give their inputs. So, on behalf of the organizing team, I sincerely thank each and every one of you who has fetched their time, precious time, to attend the meeting. So, today's meeting is about microbial solutions for environmental sustainability. And this topic itself is quite broad, and I hope you all may be aware of the uh, bio-based economy or bio-based solutions that is being promoted almost uh, in each and every domains of research, so that the whole process becomes sustainable and eco-friendly. Uh, it is in line with the sustainable developmental goal, which is quite uh, it is the call of the time now that we all should practice or should promote the SDG goals as much as possible, so that we can save our planet Earth. And bio-based solutions, especially microbial-based solutions, uh, also holds the key major aspect of it. Right, microorganisms, they are invisible, but they do or they carry out a lot of work that includes waste degradation, pollutant degradation, bioremediation, and on the contrast, also they produce antimicrobial resistance too. But we will focus more today about the solutions that microbiology can provide for environmental sustainability. So, with this thing in picture, I invite our experts and professors who have joined today. Professor Prakash Alami has also joined the meeting. Good evening, sir.
Good evening, good evening. Little bit
Yes,
busy
sir.
schedule, but I have
No,
some
sir, you are just on time.
time.
We have just begun this
Yeah.
meeting. Yeah. So, if someone would like to share their idea or their work related to this field, they can begin. Especially, I would like to invite a student participant to speak first if they want to know something specific or they are working already in this area. Today, I see Jayanta. Jayanta, are you there? Jayanta, Krishna, Prosenjit Mandal. Even Thatoi sir, yes.
Hello.
Uh, y- yes, sir. So
Are you
I think, sir, yes, yes. Then the participants, they are, I think, waiting for us to begin, and your experience is much required to for today's talk, sir. If you would like to
sure?
share
Sure.
some views on this topic about microbial solutions for environmental sustainability, please, sir.
This is a very good topic you have chosen today. Roundtable topic. This is environment is now having greatly polluted, and degraded. So, what is the sustainable solution? If we ask somebody, so it is the microbial solution that can provide in the form of bioremediation, biofuel production, uh, waste minimization, waste valorization, many sort of applications it carries. Okay. Uh, but interesting is that the microbiologist we can broadly categorize in the two groups. Some is basic research. We are all involved in academic field, basic research. But application research is very limited, very limited. But we know, all of us know that that has a great prospect. Application of microbial technology for bioremediation or um, any solution to the environmental problem. It has great prospect. So this topic is very interesting. I personally worked on bioremediation of mine waste areas, mine waste soils, chromite, iron, coal, etc. So other areas of bioremediation are organic pollutant, heavy metal pollutant, oil spill. So there are large prospect in this context. Actually, prospecting of microorganism, characterize, identify, and evaluate their application is important.
Yes, sir.
So last part is very important. Evaluation their application, then technology development, then transfer this technology to the field. That part is always very, very limit. So if we can plan, and we cannot plan this unless we collaborate with industry people or collaborate with the mining people, those are really polluting. So this is very important. So I am uh, heading a institute, Center for Industrial Biotechnology Research of Sikha or Anusandhan, deemed to be university in Odisha, which is the uh, I can say it is the high NRF ranking institute, 14th rank in India. This institute has established this Center for Industrial Biotechnology Research, where we uh, look forward this application-oriented research. So this is what I wanted to speak. Let us listen to others also, please.
Sure, sir. Thank you, sir, so much for joining the session today and providing this useful insight. I hope students or researchers have got some idea. It is quite a big term because microbial technology is emerging very rapidly, and this term microbial technology has also come in several institutes as a branch, as a new branch of like microbiology, applied microbiology aspect. So we have this microbes to degrade pollutants, to produce or valorize waste to wealth. So there can be bioenergy, like all the kind of the AB fermentation related organic uh, acids or alcohols, uh, biohydrogen, bioplastic, and if you talk about the from the remediation aspect, they degrade pollutants. Oil spillage side, they can degrade those uh, oil contents, and then they can also remove heavy metals like servo has been working in chromium reduction or mining site areas. Microbe, he has worked a lot, and he has contributed to science in that aspect. So microbes can reduce these heavy metals to make them non-toxic to the environment. Right. So this is just a small background related to the microbial solutions, and what sir has rightly suggested is that application part of this microbes are uh, were least studied, but nowadays people are trying to develop technology based on microbial solutions. One such thing that comes to my mind is one of my senior was working in South Africa. So that time they worked on microbial. They were developing co-culture of microbes that are means that are able to tolerate wide range of pH as well as pollutants, so that that can be degraded and used as a formulation to in wastewater treatment. So they got it patented, and now they transferred the technology to other companies who are using those formulation, those organisms basically as a co-culture, so that they can use. Every industry has this mandate to treat their waste within their campus and then release effluent that is already treated to the limits, to the allowed limits as per the government norms. Right. So there, these formulations works a lot, and several other teams are working on different aspects of microbial solutions. Maybe all our participants may also know some of the unique applications of microbial related applications. So I welcome all others. I will start with Arpita Joshi. Arpita Joshi, are you there? Arpita, Brijesha, even if you are a student, I think you should all participate and ask questions. Today, Professor Thatoi and Professor Prakash Alami as well as Dr. Anand is there to give the useful insight. So, and Dr. Divya is also there. Hello, ma'am. Last time we had some discussion on, I think, microbial polymer. Dr. Divya, are you there? Suhas Kulkarni, you would like to say something, Dr. Suhas. Dr. Suhas?
There are some students, they are in they were in first year, so now they will be
Okay.
in second year BSc, sir. Like Brijesha and Arpita Joshi, they all are my students. They are now they will be in second year. So I told them to join now, and maybe they don't have much knowledge about the environmental. I mean, about the microbial solution for environmental sustainability. So uh,
Yeah,
I
sure.
told them
But
to read about that
okay.
topic.
Yes, yes, yes. Yes, that is fine, sir. It is all about discussion. So, whatever the students, to researchers as well as students, we always say that that's why some briefing has been
Yes,
given so
yes,
that
yes,
they
yes,
can
yes, yes.
get
Correct.
to know
Brijesha, if
about
you
the
have a,
topic. Yeah. Uh,
we have discussed something about the environmental sustainability. If you have any question,
the conventional thing, at least students have read quite commonly, is ethanol production using yeast.
correct.
And then maybe acetic acid or ABE fermentation, if they are microbiology student, they must have read this or they will be going to read it in the coming semester. Right. Anyways, thank you so much, sir, for bringing all these students here today. It was it would help them a lot, I guess. But
Okay,
I would
I
like
have a
to.
one question, sir. Hello.
Yes, yes.
Uh, 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 Sir, actually, what is the role of, I mean, carbon sequestration? Can we include carbon sequestration also in,
Yes, carbon sequestration, mostly this greenhouse gases, especially methane, or we also call it as C1 carbon source nowadays. So,
okay,
methane or carbon dioxide, few organisms can convert them into, uh, say, formic acid or acetic acid
okay.
or related organic compounds. Okay, so that is because they are present in the gas as a contaminant only,
Mm.
like greenhouse gases. So, if they are present in our
Yes.
environment, they will raise the issue of global warming, right? And a
Okay,
lot of
okay,
industries are releasing gases that are composed of methane or maybe carbon dioxide, carbon monoxide, etc. So, if these gases can be consumed by, say, algae or any other bacteria, then they can convert this, they can use these as a carbon source and convert them into useful chemicals.
okay.
So
So
that
many
is,
keystone taxa from, like, I think, chloroflexi, some of the proteobacteria, alpha proteobacteria, acidobacteria, they all are found associated with carbon transformation process, right?
yes, yes.
Okay, okay.
So CO2 sequestration is a kind of hot topic, and even algal strains are
Yes.
being utilized nowadays to produce lipids or maybe pigments. like
Pigments, correct.
omega-3 related fatty acids or pigments like carotenoids. So these
Okay,
are quite
correct.
hot topic, and if somebody can develop a solution so that one okay, thanks for asking this one unique study. I forgot the organism name, but there is a company called Lanja Tech. Lanja Tech has
Lanzatech.
developed, yes, Lanja Tech. So in the past 10 years itself, they have developed themselves with a patented technology. The idea
Oh,
is to immobilize microorganisms and put the gas that they want to convert, this CO2 or carbon monoxide uh, containing gas, from the bottom, and they release these immobilized beads containing microorganisms from the top. So it is like a column, bubble column reactor kind of thing, where they will put the microbes from the top. Slowly they will come down while the gas is going up. So this gives more chance of interaction of these immobilized microorganisms and with the gas. So while they are migrating down, they will convert these gas molecule or carbon monoxide into acetic acid, or I think ethanol. So some fuel they are converting using carbon monoxide,
yes,
and this
yes,
is
yes.
their unique patented technology through which this company has been making profit a lot. Okay, all they got to know is this Wolfe cycle and how it is going to work, or it can be applied in carbon sequestration. So Lanjatech has collaborated with other governments also. Yes, Brijesha, you want to ask something?
Yes, sir. Sir, I have a question about oil spilling bacteria.
Mm-hmm.
Uh, Sir, how long does it take for a microbe to like clean up the oil spills? And after cleaning up, do they die or like they multiply?
After cleaning,
Yes,
the
sir. Like
no,
how
last of
long does it take to actually clean
yes,
up? And
actually yes, the microbial process is relatively slow, but I hope you have heard about the organism Pseudomonas putida. Pseudomonas
Mhm.
putida is one of the organisms that can be used for such process. Even there is an institute in Gurgaon, that is TERI. So Dr. Banwari Lal has developed microbial co-culture. They call it oil zapper. Okay, they have nicknamed that consortia as oil zapper, that they are supplying to Gulf countries to be applied at this such kind of oil spillage site. Although microbial things or enzymes work slowly compared to chemical based methods, but it is environmental friendly, and slowly ultimately it all means it consumes or it degrades all the oil spillage, so that the environment can be protected, right? So the process is slow, but it happens in a very eco-friendly manner.
so after degradation, does microbes like die or do they keep multiplying?
No, they will keep on multiplying, obviously, and they are not harmful for the environment. So even if they are there in the environment, unless it is Pseudomonas aeruginosa or some kind of opportunistic pathogen, then only it will become a issue. Okay. I think Tatoi sir want to say something. Tatoi sir.
Yes, it's a super bug. There is a Chakravarti, our
Yes,
famous
Anand Mohan Chakravarti sir. Yes, yes, sir.
Chakravarti,
Yes,
uh, given the name superbug to this oil-eating bacteria
superbug.
or oil-eating bacteria, right? That is very, that is the first time it is actually genetically engineered bacteria, I think. That was discovered, and it removes all the oil spills, can remove all the oil spills, and it is not harmful to the environment. It is not at all harmful for the environment.
Yes, it was Pseudomonas putida strain, and I think toluene degradation pathway they cloned. So
Yeah,
related to
yeah,
toluene degradation things he cloned, and a lot of plasmids were incorporated in the same organism so that it can degrade multiple pollutants at a time. That's why it
yes,
is named as superbug. sir, as rightly said that.
Many genes or plasmids are introduced through genetical engineering protocol, so that these bacteria are very, very efficient.
Yes. So if we consider that as a general term, if a bacteria can consume only one type of carbon source, so its food is limited. Through genetic engineering, we can allow them to enjoy multiple different types of food. That means different types of chemicals they can metabolize and grow using those as carbon source.
Another area I want to focus, that is the plastic degradation. It
Yes,
is
sir.
a
Yeah.
great menace to the environment, and there are many technologies already developed in place. You can find many patents have been developed. I have also filed one consortia developed against the plastic degradation. So that is a very good research field actually nowadays, and government is uh, funding if somebody wants to study plastic degradation.
So
And there are
rhodococcus is also, I think, for bioplastic, we are using rhodococcus rubber.
no, there are two different aspects actually. One is bioplastic because that is the
Hmm.
resource we are going to develop
Okay.
from the
Hmm.
microorganism or any other source. Another thing is the degradation, uh,
Okay. Yes.
bioremediation of the plastic,
Hmm. Hmm. Hmm.
waste degradation. So that is also many microorganisms they degrade the plastics uh, very in a very short time. Short time, maybe within 6 months, plastics can be degraded. You can pile up the plastics and put
Okay.
the inoculum over it, and it will degrade very easily because plastics remain in the soil for many years, for more than decades, so it can easily be degraded.
Yes, sir. Here I would like to ask you, sir, Tatoi sir,
Yes.
when we talk about plastic degradation, how we feed bacteria this plastic means we need to solubilize plastic in some kind of organic solvent, or we just give a solid substrate as it is.
No. Only initially you have to grow microorganism in a carbon in a carbon medium actually suitable for its growth. Then we can add plastics to it, and they secret actually enzymes. The mechanism is they secret enzymes, certain enzymes. That enzymes actually degrade the plastics. They break down these plastics actually. That is the mechanism. And it is
Yes,
a
sir.
very
This.
good research field. It is a very good research field. Students
Mhm.
should start working, and it will draw attentions. That is my intention to tell students that they should work very easily. Isolate the plastic degrading bacteria from pile up from the environment where plastics are thrown up. From that environment, they can isolate, they can identify, then they can uh, carry out certain small experiment in the laboratories, and then study uh, how the plastics, the plastic degrading ability of those microorganisms.
Yes, yes. PETase, that enzyme PETase, it was first recognized as means the organism. I forgot the name, but it was a huge talk of the town that time when it was first recognized and identified. And based on in silico analysis, also people have started creating clones of PETase so that the substrate uh, enzyme, so the substrate level can be increased. Means different types of substrate, this enzyme can degrade. So there are several studies going on, while people are also looking after degradation of microplastics, as the microplastic presence in uh, water bottle to rainwater to even ice bugs are even in our blood microplastics are there. So people are trying to identify the presence of microplastic using unique stains, as well as degrade those microplastic using uh, Microbial solutions.
Yeah.
So, sir, your patent work, would you like to discuss or give a bit brief about that? What was the work or how you actually isolated a strain that degraded? It would be helpful for the students, those who would like to carry out 6 month or
The
1 year project.
same process I told from the municipality waste heaps where the plastic were thrown. Actually, from that environment we isolate. We brought the soil, then put it into some culture media. Then we put the pore plate and found the colonies and pick up certain bacteria which grow uh, efficiently in that medium. Then we identified them, then again evaluated their plastic degradation process. These are the certain protocols are there.
Yes, sir.
But it is interesting, very interesting.
Yes, actually, this plastic waste problem has become like it is not going to fade away soon, it seems, and microwave
Yes, yes.
solutions would be helpful, very much helpful.
Yes,
Good
yes.
evening, sir. Good evening, sir.
Yes, Jayanta, tell me.
Sir, uh, I, I completed PhD in microbial consortia. So, question like sir said that there are many bacteria who are able to degrade plastic, but there are many report also for fungus. Like most of the studies say that fungus have more better ability to degrade plastic and some complex compound and other bacteria because they have good lignocellulose activity and other related complex compound. So it could be better than bacteria then.
I don't have a idea actually that part. Whether fungus are better plastic degraders or the bacteria. But yes, in so far the enzyme production ability, the fungus are a very good enzyme producer in comparison to bacteria. So that might be the reason that fungus can be better. But what kind of enzyme they have, that literature review actually I have not done, so I cannot speak it now. Mm, because I
And then one
have.
more problem. Yes,
Hmm.
yes, yes. And one more problem is that majority of the plastic available are synthetic plastic. Like biodegradable plastic are trending now. They are emerging in many areas slowly.
Hmm.
But
Hmm. Hmm.
majority we found the plastic are more synthetic, like chemically synthetic polystyrene that. So microbe have an enzyme that have could an enzyme for degradation or something pathway.
Uh, there are different types of high density or low density polythene or plastic are there. So depending upon that, the degradation capacity and ability, and again one microorganism or consortia microorganism, and their pathway definitely are different for different microorganisms. So those things are the somebody is expertise he has already carried out. Only I studied this as a part of one of the students, PG students. So I have not gone detail into that. But
Gemma Marketer.
that is the field exposed to the students. Students should study the literature. They can explore those those things, and you can supplement if somebody really worked detail into those aspects.
Yes, and I think I read somewhere that regarding polystyrene, I have read that bacteria, some of the bacteria are identified uh, to degrade polystyrene. Okay, styrene monomer. can be utilized. So I think it was Pseudomonas or some related organism that they isolated for that. But obviously, plastics are a bit complex. Not all the enzymes can access. And regarding your question, yes, fungus secretes a lot of enzyme, but mostly they are used to valorize or convert lignocellulosic biomass into various products. So, like cellulose can be converted to glucose and then be used for biofuel production, biobutanol, SAF, sustainable aviation fuel, like those things. Okay, or fungal enzymes can also degrade lignin. Okay, capacity varies from organism to organism. But fungal enzymes against plastic, I think I have not read at least, but it is quite interesting. Maybe people may be reporting this also. So thank you,
Yes,
Dr. Jayanta. Yes, if you
and
are working,
actually,
yes.
yes, I'm work on cellulose degrading bacteria during PhD. But the question in bacteria are, or microbes are very easily grown in lab because they have very easy carbon source. But when we try to make them in field trial, they mostly there are many different challenges. Like because in laboratory condition we provide all optimum condition like accessible carbon source. Like you say, some microbes are not able to get the is accessible carbon source from directly environment. So other microorganisms are cooperate them to solubilize that carbon. So then they uh, start their own functioning. Like this because like I want to degrade the cellulose degrading bacteria from culture enrichment techniques. When I tried in the rice husk or straw like substrate, that very complex and consistent with lignocellulose, different organic complex materials. Then that time they have difficult to solubilize this.
Yes, yes. That issue is a bit miss. If I understood correctly, it is a bit different because in the lab we either use CMC like carboxymethyl cellulose or alpha
Yes.
cellulose. So pure substrate we are using, and while we are targeting lignocellulosic biomass, so even rice straw, wheat straw, cotton or bagasse, anything you talk about, they are mixed with lignin, and lignin makes this process of degradation a bit tedious for the enzymes because enzymes cannot access enzymes we worked on in the lab is cellulase, while these cellulases has to pass through lignin, which is a kind of recalcitrant compound. So if there is no leakage or means lignin degrading type of ability, then it will not be able to degrade. And to overcome that, we need to go for pre-treatment. So pre-treatment of the biomass is important before applying any enzyme or enzyme releasing fungus or bacteria. Okay, so through pre-treatment, we remove the lignin component. So now the cellulose is free or accessible. Even hemicellulose is also free after pre-treatment. So we need to choose a perfect combination of pre-treatment as well as a microbial treatment to get more and more sugar released from the process.
Yes, sure.
So you may have heard of steam explosion. Uh, there are acid pre-treatment method. There are even alkaline pre-treatment method, and so there are so many combination of treatment as well, even ultrasound and so many other things. So people try to basically remove or loosen the lignin binding from this lignocellulosic biomass. But
Yes, it will give better result. And like I tried in autoclave, like when substrate autoclave and after autoclave, it's very easy to convert that substrate into the simple sugar.
yes, yes. And the only lacuna of conversion of lignocellulosic biomass is the pretreatment only. The cost of pretreatment as well as cost of enzyme. These two things are a kind of hindrance. So once optimized properly, it will be helpful. And another thing that you rightly said is not a single organism, but people also try to develop co-culture. If you talk about the uh, success of a culture in a actual environment or field environment, then uh, as bacteria don't work alone in the environment, so people try to develop co-cultures so that they efficiently degrade the waste, right? Let it be plastic or any other thing.
And there are many other microbe also reported that someone says about carbon sequestration. Like many during the waste degradation, there are many odorous compound emitted in the gas, like mostly the methane gas or H2S or some volatile organic compound. There are many bacteria have transformed that gaseous compound into the other compound. They
Yes,
control the odor emission. This is also one of the emerging topic in the microbes for odor control.
yes, yes.
Like mostly in mostly the reported study are China because the China has degrade the manure, animal manure, and most of the manure degradation emit the very bad nauseous odor, which dislike people to go for the treatment. So it's also one of the area which explore.
Yes, yes, correct, correct. And in this connection, one unique thing that I got to know is through lignin degradation, we can generate vanillin. Vanillin is vanillin, it is having very good kind of odor. So, like a perfume. So, from lignin, people can make perfume. Although yields are not good, but such kind of uh, work is also going on. Yes, you are very right. Great point there.
And vanillin is also a quorum sensing inhibitor, I think. You know?
Okay, I have not heard of it. Okay.
Actually, it is a quorum sensing when bacteria communicate through quorum sensing mechanism. So the vanillin are most widely reported quorum sensing inhibitor or
Mm-hmm.
many. Actually, bacteria in the environment
AHL,
communicate.
AHL degradation is there, Dr. Jayanta. AHL degradation, acyl
Yes,
homoserine
yes,
lactone, right?
yes, yes,
Hmm.
yes. Yes. Well, like mostly the monitored to the HAHL or some are other antipeptide like, but vanillin are inhibited this mechanism very well. Many research are reported this.
Mm-hmm. So yes,
It is useful in the bio control agent, like in biomembrane filtration process. Biofouling are happens due to accumulation of organic particles on the membrane filtration. So that area it is very useful. Vanillin.
yes, yes, yes. For the students, this is a very unique aspect about biofilm. So, biofilm, already we had a discussion in some other roundtable meeting, but biofilms are good as well as bad. So, this is regarding the bad biofilm on membrane bioreactor or any kind of membrane filtration process if you are doing. So, bacteria will keep on accumulating and growing on the membrane and they will choke the membrane. So, it remains like underutilized. So if we can degrade the biofilm formed on the membrane, then the life of the membrane as well as the whole machinery will be miss more, right? So there vanillin can have function. Yes, thank you, Janta. Just I want to interrupt because there is a chat message by Oshi Biswas. She or he, I don't know. They want to discuss about microbial fuel cell and how they work and what is the purpose.
I think we can microbial fuel cell environmental में discuss करना. I think मुझे लग रहा है. I think there is some different topic on microbial fuel cell. Environmental
Uh,
sustainability we cannot.
yes, yes, but just to satisfy the curiosity of Biswas, yes, microbial fuel cells are also being worked out where we used microbes in either cathode or anode position and produce biohydrogen or organic acids as an output. So microbial fuel cells are very much utilized in wastewater treatment process. Okay, or even utilizing volatile fatty acids present in the wastewater. So whatever carbon source available in the wastewater, that can be converted to either electricity, hydrogen, or other organic compounds through microbial fuel cell. So just fuel cell, when you say that it is a fuel cell, so mostly it is about generation of electricity using bacteria or bacterial consortium. But otherwise, the broad term is bioelectrochemical system. So that also covers the part that is called desalination. So, Biswas, if you have any specific question, you can ask. We will try to answer. Or I hope you had the flavor that the bioelectrochemical systems are utilized for electricity generation, hydrogen production, or organic compound production, as well as even in desalination program. So you can basically remove salts from water. So in the Gulf countries, people are trying to create uh, microbial fuel cell or desalination thing using uh, uh, microbes. So there are small reactors people can connect and that can be used. So at large scale, it is so microbial fuel cell works like a battery, small batteries connected together in chain, so that net output can be very high. Okay, current limitation is the amount of energy being produced per reactor. So we cannot use a very large size of microbial fuel cell. So, and the cost of the membrane that is used, proton exchange membrane in a microbial fuel cell, it is also quite expensive. So people are trying to develop a cheaper proton exchange membrane so that the cost of the microbial fuel cell goes down. So, Viswas, are you there?
Yes, sir. Thank you, sir.
Okay, great. Tell us something about yourself. You're a researcher or a student.
Sir, I'm a student. I was curious about learning more, so that is why I joined this discussion.
Very good. Yes, yes, it is about discussing things as well as whatever things you are interested in, because mostly it is up to the students now that they will carry out the research, they will get interested more on different microbial solutions. They will try to develop applications related to microbial strains. Let it be enzymes, maybe pesticide degradation, maybe nowadays people are also working on antibiotic degradation from wastewater, like say Yamuna River or any river is getting a lot of sewage. All the sewages are also having a lot of antibiotics. So antibiotics level in the river or water bodies are accumulating a lot. So if there are microbes or if there are some kind of consortia that can degrade those antibiotics, that will help reducing generation of AMR in the environment. Okay, antimicrobial resistance bacteria can be generated or produced through mutations. So if there are more antibiotics in the environment, they will get resistant to it. Right. So if we degrade the antibiotics, we will lower the chance of getting or generating AMRs. So that is the logic of degrading uh, antibiotics from the wastewater or water bodies. So anyone else? Today I have not covered. I think Jyoti Thakur, are you there? Jyoti, Katha, Musarrat, Prasenjit.
Yes, sir, I'm there.
Yes.
And
Please tell us something about yourself and what are you working on?
sir, I'm Prasanjan. I am a PhD, first year PhD student.
Okay.
So I am working on microbial volatile organic compounds.
Okay. Volatile organic compounds. So you are converting them to some product or you are producing VOC?
No, no, sir, I want to see how the microbial volatile organic compound affect the plant's metabolisms, how they affect the plant's secondary metabolism
Okay.
or the yes. So sir told that the microbial volatile compounds, as we know that the microbes produce microbial volatile compounds. So can that many bacteria can some type of bacteria can produce the scent like uh, as sir already told that they can produce perfume also one of perfume like volatile compounds. So
Yes.
some strains or uh, is available or is there? I want to know for
What strains are available or not?
yes sir, yes sir, yes sir.
Yes, yes, they are available. From microbial culture collection, you can get some of them, not all. Yes.
Okay sir.
I hope you know about actinomycetes also, no soil bacteria that produces this the typical rain smell that we
Then
perceive. Yes, yes.
pseudomonas.
So those are actinomycetes that generate
Yes.
these compounds, right? So there are several other strains also. Uh,
Mhm.
Even for like lignin to vanillin, people have identified fungal strains and even chemical process also to convert lignin to various other ferulic acid uh, as well as lignin, vanillin related compounds.
Geosmin, by actinomycetes.
Actinomycetes, no, yes, yes, they produce juicing.
Geosmin, yes.
Jasmine, sir, yes. Mm. After the rain, the actually the scent for this. Yeah.
Yes, yes. So great. Thank you, Prasenjit, for joining.
Thank,
Uh,
thank you, sir.
Rigved, Reena, Upasna, are you there?
Yes, sir, I'm
So,
here.
yes, Upasna, please tell something about yourself as well as what are you working on?
So I am currently working as an assistant professor in this Assam Royal Global University here in Guwahati, Assam, sir. I am in the Department of Botany, but my specialization from BSc itself it's microbiology, sir.
Okay,
So
great.
I have worked on this river ecosystem where I have studied this heavy metal pollution and the co-occurrence of antibiotic resistance in these heavy metal tolerant bacteria in River Tista, which is in Sikkim. So that
Hmm.
was a part of my PhD, sir, and currently I'm pursuing my research, continuing it in pesticide degradation in this from the bacteria, rhizobacteria isolated from tea gardens of Assam. So I have a PhD student who is working on currently, just recently he has joined. So this is his topic of research, sir.
Great,
So
great. So
yes, sir,
that
because
part,
pesticide pollution is also very concerning, sir, when we talk about this agricultural field or agricultural ecosystem. It's building up and it leads to the degradation of soil health, this what we call as microbial dysbiosis, and that is ultimately hampering this plant growth. So we are looking into the part that if we can also work on any bacterial consortium that can effectively degrade pesticides that are widely used in these tea gardens. So that is my aim.
okay, great, great to learn. Yes, even Himalayan bioresource are too much. All the Himalayan region, including that part, no, northeastern region, has a lot
Yes,
of
sir.
biodiversity that is yet to be explored.
Yes, and even a lot of work has been done here in this coal mining region where this the water that leaches out the nearby water bodies are very much acidic. So my own guide from this Northeastern Hill University, they have worked on this coal mining remediation of this coal mining water bodies,
Okay,
and
great.
they have also, yes, developed a very sustainable or eco-friendly, a very simple technique to remediate that water, which is actually highly acidic and having a lot of iron in it. So
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 it was
if
a
you have
bacteria that
experience,
they used, or it was some kind of
yes, sir,
consortium.
it was a bacterial consortium only, sir.
Okay. Hmm.
So, so have you any experience working in pesticide degradation?
No, but I think Thatoi sir work is quite related to yours, no, regarding bacteria present in mines as well as pesticide degradation. I have never worked on these topics, but yes, I have read about it. Uh,
Yes, sir.
because selection of the pesticide is also important, which reason you are choosing. Thatoi sir, you
True.
want to say something, sir, please.
You are talking something mine waste degradation. Okay.
Coco, please.
Okay.
Sorry, sir, someone
Okay.
just
Okay. Now
joined. Yes, sir.
you are talking something mine soil degradation by
No, sir. Actually,
microorganisms.
pesticide degradation, sir.
Pesticide.
Yes, sir.
Yes,
From the
Dr. Upasna want to know if you have worked on pesticide degradation.
No, no,
degradation.
I didn't work on pesticide degradation. I don't think pesticide degradation most people have done on algal flora. Are you talking about algal pesticide degradation or bacteria?
Bacterial, sir. Bacterial.
Bacteria,
Rhizobacteria, sir.
rhizobacteria. Okay,
Yes.
I don't have much idea. Mhm. But I know
I feel there is a
pesticides.
microbial technology. Yes, sir. Sometimes it is like there are so many literature on pesticide degradation. People have worked on single strain or the consortium, but I feel there is always this gap when we talk about field study. Uh,
Yes.
so the
This is
translation
about
of technology
algal
is
flora you
very,
are
yes.
talking, no?
So, like any microbial solution, when we talk about bioremediation, any like bioremediation of heavy metal could be or pesticide or hydrocarbon. So in these kind of studies, I feel like sometimes there are many good studies which is just limited to the literature. So it is not getting translated, or maybe there is some challenging part that is there where we are not able to translate this into actual field when we talk about actual in
See,
situ bioremediation.
I just wanted to tell you certain aspects of the uh, application part. Microorganisms, yes, isolation is very crucial, and isolation always
Yes,
done from the environment where the toxic or these pollutants are present. And because this
yes,
microorganism has developed certain capacity or tolerance to such environment, and they are able to grow. It is the beauty of this microorganism is that they are able to thrive at any environment, whether bottom of the water or inside the soil or in atmosphere or anywhere they can thrive. The reason is that they have unique capability of producing certain enzymes. So metabolic
yes,
activity for their own benefit, one growth and development, they carry out certain metabolic activity with help of this enzyme. And those enzymes, they when they adopt, they usually uh, develop tolerance or resistance to such. And we are searching those microorganisms which are resistant. So obviously we will search for those environment. We will explore those environment the for the microorganisms. Then we have to isolate. Then we have to make pure culture and store. That is one part. Second part
yes,
is going to evaluate their ability and their the nobility. You have to study their nobility and their ability or efficiency. If somebody is very good, then it's okay. Then there are options whether we'll use native microorganism or go for the any engineering part. Why engineering part is required? Because native organism has a limitation. If you want to
Yes, sir. Yes, sir.
more proficient, more potent, then we have to go for the engineering path.
Engineering.
And
Yes.
that required cloning. That is the cloning path.
Yes.
So when we do cloning, we have to insert the required gene to certain other very promoter with certain promoter introduced to certain other microorganisms. Then it is the enzyme production ability becomes very high. Then we can go for the application. So before that,
Yes, sir.
we have to any application they go through the fermenting process or where the fermenter is used, and then the engineering part, the fermentation always associated with the optimization process. So optimization, fermentation,
Yes, sir.
those are associated with your uh, upscaling. Okay, then you can go for whether enzyme or the microorganism, anything, you can go for the application. So before application, lot many things are
Yes, sir.
required.
Yes, yes. True, sir. Thank you, sir.
Sir, Tatoi sir, and Prakash Alami sir, if you are there, this one question was always there to ask in the roundtable discussion is about, say, I am I have isolated a environmental bacteria from uh, any soil. So, means uh, it is a Indian territory. So, we have to go for NBA always, or is it that we just submit the strain to a culture collection agency, and then we can start working on that isolate? So that NBA clause, I'm not much clear. So if you can shed some light on that.
NDA,
NBA,
non
yes.
non disclosing.
No, sir. National Biodiversity Act, na. I
No, no,
think it is
for bacteria, no,
about
no, no,
utilizing biodiversity, natural
no, no,
biodiversity for
no,
research.
no. For bacteria, I don't think that is required. Bacteria, there is by resource is not required, clearance is not required. It is for the plant, animal, and all. We don't require for the microorganisms. You can go directly. You can submit. Okay, if you are going for the development of
Patent,
patent,
yes, sir. Patent
then
related. Yes.
you can submit. But there, the submission should be not for public. You have to give a fee. You have to provide a fee to Imtech. They will preserve your strain, and they will not share it with anybody else. Only they
Okay.
preserve and give a MTCC number. That's all. But if you want, let others also use your strain, then you can, without deposition, you can freely deposit your strain, and you can get a MTCC number. You can work and you can publish. You can also patent. There is no problem for that. But if you want to have an exclusive right with you, then you have not to public it. So that is the process.
Okay. Okay, sir. Lastly, Dr. Prakash Salami, sir, are you there, sir?
I
Jayanta,
would
you
like,
want to say something? Yes.
Upasana is here.
Yes, sir. Yes, sir. I'm here.
You are trying to for bioaugmenting rhizosphere or biostimulating? Which approach you are utilizing for soil?
Sir, actually, till now I have not gone up to that part, sir. I am just till now, sir. I have just isolated. I have got the isolates, and I am screening those isolates for the. In fact, right now, just tolerance to pesticides. Like I have used thiacloprid, I have used chlorpyrifos. These pesticides. So then after that, I will select the isolates and I'll go for degradation part. And my aim is to develop a consortium, and then yes, sir, it will be bioaugmentation only. I finally, sir.
Actually, you so got idea. There are two approach. Mostly that to bioaugmentation because we are
Yes, sir.
adding the that already one microbial community existing in that soil.
Yes,
And
sir.
most of the lab study not translated into field study because many of the study are only limited in the microcosms level, like they done
Yes,
in 5 kg or 10 kg. But when we need to translate the research that identify the challenge, we need to go for pilot scale study because most of
okay.
the time in PhD study, even my no, we don't go in that level. Because pilot scale level need
Yes,
more quantity of waste sample, more team power, and when we are alone, researcher independent, it is very difficult to go for that study.
yes, yes,
And
true.
more preferential if the already if you got know about the sequencing and microbial profiling of that soil where the pesticide is available, it many high chances that many microorganisms have potential to degrade that pesticide may be present in that soil already because adaptation are
Okay.
going in many
Yes,
microbial ecology now. So it's
yes.
preferably if you stimulate that microbe, it will more beneficial because they already known and adopted on that soil environment condition. Because
Okay, yes,
bioaugmenting
yes.
microbial cell are sometimes compete the other microorganism in that environmental field,
Yes,
or
yes.
sometimes they
Competition,
fail.
yes.
Even they have potential of degrading that pesticidal gene, but because of due to unfavorable microbial competition is there, we need a pilot skill study like a real sample, like you take the whole soil sample that contaminated because the pesticide level is also differ.
Yes, that is also
If it
again
is
true. Yeah.
10 ppm and we need we laboratory study, we mostly go for 1 ppm or 2 ppm. That's also matter because a very high pollutant toxicity is also affect in this. So it is systematic approach. We need to microcosm, mesocosm, and pilot skill study. Then we can translate gradually from this to this.
Yes, yes. But yeah, these researches like these work should be gradually developed. But at just at a PhD level, I think a student will not be able to achieve everything. So, but this
Yes,
can
yes, that is
definitely be
the challenge. Yeah,
completed.
I
Yeah.
understand it very well because it's not possible
Yeah.
to cover up in one study. And many study are focusing
That
only
he gets or whatever
most.
outcome
Yeah,
from his
I
work that can definitely
even
be.
even I experience many study focusing on isolation and
And
microcosm study limited. But we
yes,
have to
yes,
need to more focus because many my phylogenetic. If we read the phylogenetic, many microorganisms already reported. We have to think about the beyond the next level.
next. Yes,
What
yes,
is the
true.
behavior of microorganism in pilot scale study, like column
Yes,
study or whatever study is feasible at or laboratory?
Yes. Thank you, Janta, for your valuable input. And
yes. Thank.
yes, Prakash sir, you came online. Just some concluding remarks, and if you can inform something about NBA, how can we go with that?
Yeah, definitely. I think today's discussion was really wonderful, though we had a little bit less attendance. Now we are ending only with some 12 people. But the topic which was covered, the pseudomonas potato degrading hydrocarbons, the superbug, then plastic degrading peptide enzyme of producing organism, as well as the discussion from the different aspects, either to use as a consortia, microbial consortia, bacterial consortia, bacteria versus the fungi, you know, and all actually are very much required for the sustainable development or to achieve the sustainable development goal. And I think microorganisms are the microbiology is one such a important subject which we can always use as a renewable source. As a, in the bio-E3 concept also, we have several such activities with the DBT, where I am associated with bio-E3 projects of the DBT. And microbiology, I think, plays an important role when we see all these aspects. So from that angle, most of the students also participated very actively. Coming to NBA, I think we should have one of the seminar, not directly talking about the NBA, but patenting the organisms, then their utilization, the culture collection centers, and then especially when we are, sending overseas, because when we are sending any biological
Material
material,
transfer agreement, yes, sir.
you know, even as a for the research purpose or for the industry purpose, so there are actually the different procedures involved. Because whatever we have the biodiversity material is not our property, it is national property. And government of India actually following such strict rules how to utilize that. So biological material assets cannot be transported. You get even the biological material from outside country without proper declaration or the permission, it will be discarded. So maybe not directly talking what NBA should do, but what are the procedure we should follow related to that. I think we should have one such roundtable discussion so that it will actually will be quite useful, because right now we are also in a process of sharing such a few bacterial cultures now outside for a research purpose. So what we are following is only the material transfer agreement and all. Or when you
Yes, sir.
get the cultures from the culture collection center like ATCC is there or DSM is there, you know. So we are actually purchasing those culture and we are actually declaring what is this biological material is used for, right?
Yes,
But
sir.
NBA, which is actually headquartered in Chennai, is playing a very important role. Though one or other problems or difficulties also will be there while exchanging such material, but important for us to follow all those procedures. So maybe related to the Biodiversity Act, even we also organize one seminar
Yes, sir.
on the
Anand, sir, we will keep this as a note, and next meeting,
state,
some meeting will be. Yes, sir.
State Biodiversity Act, then National Biodiversity Act. So it applies to all the biological material, and how we can actually be careful while sharing the material, utilizing, and all these things. And as you know that India has several such biological hotspot, biodiversity hotspot, and many of those are not actually explored. Whether it is a Himalayan or Western Ghat or the coastal regions, these are also actually very important to isolate several new either microbial cultures, thermophiles or extremophiles, alkalophiles, all. So important, of course, few of this institute has there several such specialized culture collection centers. For algae also they have, fungi also we have, dairy cultures we have, you know, and all industrial for microorganism associated with industrial application also. So all these things are there. As of now, we actually approach a culture collection center also. They will not give you easily. They will tell to meet the means contact the original researcher. You know. So those kind of limitations are there. And most of our microbiologists actually they start isolating, screening, characterization, and then spend a lot of time in that, rather than
Hmm.
directly using the culture which is already isolated, obtained, and available in culture collection center. So I think we have to also a little bit modify our research strategy. Always isolating from natural source is a good, but then still the time which you are actually giving, and then if your objective is not screening and characterization, then you need not to look to that extent. Perhaps looking more in the advanced way, right? Just to add about the peptide enzyme, though it was published in Science, I think 2010, 12 somewhere. But then afterwards, we have not seen any much update
Many
about that.
progress, much progress. Yes, sir.
You
And
know,
I was thinking, although some
exactly,
in silico work is going on, that how to basically make it recombinant and
exactly. So we
utilize for
really
other.
don't know to what extent it is successful. Otherwise, by this time, I think our white pollution problem would have been settled, solved. You know,
Yes, sir.
now much more biodegradable plastics are available. You know, to use. Even the polylactic plastics are available. You know,
Yes, sir.
plastic the carry bags with the other biological material also it is available. Of course, plastic problem is there, but then still somehow we have not seen much development in that area. Maybe some of these experts, those who will be joining, they can also see sometimes in the literature. It will be really a good knowledge sharing. So thank you very much. Actually, a little bit busy schedule for me this evening. So that's
Yes,
why.
sir.
But
But
then I
still,
think
sir,
I
your
thought.
presence always add value to our meeting. Thank you so much, sir, for your time.
Exactly. We also used one or two just to quickly tell. I think I would have already shared also, especially in the no dehairing, in the leather processing. So rather than using the calcium chloride for dehairing, actually we use the lactic fermentation only directly. No, to minimize that calcium chloride which is used, you know, before discharging to the sewage, we treat that one we actually no, deactivate that calcium chloride, you know,
Mhm.
so using the lactic acid bacteria. So that was one process which we used. Another one again in the slaughterhouse waste. No, slaughterhouse also a lot of waste is being generated. So that actually we use directly for the lactic acid fermentation, and we could recover polyunsaturated fatty acid as well as protein. You know, that can be used for industrially or for the as a poultry feed. You know, so such. If you see that how where are the microorganism can be used for the fermentation or for treating the effluent. From that perspective, I was just wanted to tell these two examples. Okay, thank you very much, everyone. It was a nice discussion. That's all from my side. Thank you.
Uh, Thank you so much, sir. And thank you to all the participants who have devoted a lot of time. Special thanks to Professor Prakash Alami, sir, Satoi, sir, Anand ji,
Thank
and
you, Robin.
Dr. Upasna for the useful discussion. The feedback link is provided in the chat box. Participants are requested to fill it before logging out. So once again, thank you, everyone.
Thank you. Thank you.
Thank you, sir. Thank you, Prasun sir.
Yes, sir. And
Thank you.
Thatoi sir, yes, Thatoi sir, please, we will be looking for you in our next roundtable discussion as well.
Sure,
It was
sure,
nice
sir.
to have you today. Yes, sir.
But why, sir, it was a very nice lecture. And whatever information that you provide, it was really very good. I'm from Gujarat, affiliated to Gujarat University College. And
Okay.
yes, and my working area is agriculture microbiology, biocontrol, plant growth promoting rhizobacteria, actinomycetes, and transcriptome analysis of from Fusarium modum pigeon pea wilting infection.
Yeah. We are also working on biofertilizer
Oh,
formulation and
that's great.
development, all that.
Prasun sir, we will keep one lecture of, we will keep series of agriculture microbiology also. Some plant, I already talked with Ibran sir regarding plant microbe interactions and some agriculture topic also. So we'll put that topic also, sir.
Sure,
You will
sure.
be in touch. This group is very good, interesting. I really missed uh, uh, from the last year. I was in this group, but I didn't attended such roundtable or any other meeting. So I missed a lot from all of you. We are in the stage scheme. You young people are very good in the new as technology, new as
Prakash
subject.
Alami sir was the person who, for after finishing PhD thesis presentation was there, and he was the person, right, sir? Prakash sir.
Yes, sir. Yes, sir. Yes, sir.
Here, sir, Prakash sir has given a very good concluding remark. And regarding this
Super.
Thank you.
biodiversity, this thing, I was also very interested to know. I don't have much knowledge whether this. I have something has been clarified to me, but more we need maybe this issue biodiversity related issue. We can have a special lecture from Prakash sir.
Yeah, possible. We can have a roundtable discussion only, so that
Yeah,
everyone actually share their views and opinion.
yeah.
Oh,
That is very important. Very important.
Yes,
Okay.
you know.
mm. Okay, okay, sir. Thank you so much once again. Good night to all. Yeah.
Yeah.
Good night.
Thank
Good
you.
night. Good night.